We'll finish the lecture on antimicrobial drugs by completing Chapter 20. Agents can also affect fungi as well. Fungi is a microorganism that can enter into the host as well. Antifungal drugs are agents that affect fungal steroids. In a fungal membrane, the principal steroid is ergosterol. In animal membrane is cholesterol. This forms a basic selective toxicity that antifungal drugs can work at. Amphotericin B is the most commonly used polyene antibiotic. Their activity is based on the damage to the plasma membrane combining with the membrane steroids. Amphotericin B is used for systemic fungal infections. You should be familiar with this agent. Imidazole is an antifungal agent such as meconazole and chlorotrimazole, which you see in topically applied antifungal therapy, very commonly used over the counter for jock itch, athlete's foot infections, ringworm infections. These topical applications of imidazole, metoconazole, and or chlorotrimazole are used again as topical preparations. Ketoconazole can be taken orally, substitute for amphotericin B in many systemic fungal infections. Triazoles such as fluconazole and itraconazole are used for the treatment of systemic fungal infections. Fluconazole or diflucan you see commonly used to treat vaginal yeast infections. It's now sold very commonly as one-day preparations. It can be administered orally for 1 day for the treatment of vaginal candidiasis. There's also topical preparations that can be administered as well. Voriconazole is a newer azole, has a broad spectrum ability to penetrate deep tissue recesses in the body. It can also penetrate the blood-brain barrier, which it makes it effective in treating many of the fungal infections that can invade the brain, Cryptococcus for example. The alanamines are a recently developed class of antifungal agents that inhibit the ergosterol synthesis in a different manner than we see in the other azoles. Terbinafine and naphthene are examples of this. These are the agents such as Lamisil, which again you see popularly advertised now for the treatment of antifungal infections. So you have polyenes, such as category of amphotericin B, and the azoles, which were commonly administered, and the alanamines, which are used as well for antifungal therapy. There are also primary agents that have selective toxicity for beta-glucans, which are unique to the fungal cell wall. And again, as you look at this selective targeting, we're looking at agents that are different inside the invading microorganism that's different from the human cell. If you can attack a different structure in these cells, something that's far different than what's in the human cell, you stand a good opportunity of being able to destroy that agent without affecting the host cells. New classes of antifungals are used as well. Cannidines interfere with the synthesis of glucans. There's also capifungins, which are used as a trade name of Candidus, is popularly used agents as well. You can see these agents, especially Candidus, is popularly treating Candida infections. You also can see it used in the treatment of pneumocystis carni, which is a pneumonia that affects patients diagnosed with human immunodeficiency virus. Because the immune system has been compromised, these opportunistic infections can grow, particularly fungal infections. Pneumocystis carni is one that particularly is aggressive in the lungs. Griseofulvin is another fungostatic drug. You should be familiar with that name. Taken orally, this drug binds selectively to keratin in the skin, the hair, and the nails, preventing fungal growth at these sites. Tonaltate is a topical preparation alternative to meconazole in the treatment of athlete's foot. Again, griseofulvin, tonaltate, these are used to treat fungal infections. Griseofulvin popularly used for superficial mycosis infections. Tonaltate used, as you can see, as an action in athlete's foot. There are also antifungal agents that can be used to inhibit nucleic acid. Analogs to cysteine, which as you recognize is an important nucleic acid. Flucysteine is an analog to cysteine, and thus will substitute for cysteine, and thus will interfere with the process of RNA synthesis, and therefore interferes with protein synthesis. Undecyclic acid is a fatty acid antifungal activity. Pentamidine is a highly effective antifungal agent that's used in the treatment of pneumocystis carni pneumonia. Pentamidine, a little different than what we see in some of the other agents, is administered via an aerosol apparatus, so the person actually breathes this into their lungs to eradicate pneumocystis pneumonia. Again, flucysteine, these are cysteine analogs. Pentamidine, treatment in the anti-pneumocystis carni. There are a number of antiviral drugs that are, compared to that of the antibacterial drugs, very limited. Drugs used to treat influenza, amantadine was the first licensed, is thought to have a mode of action was unknown. The most new antiviral are directed in the control of HIV disease, and we talked about these previously when we talked about HIV drugs. Other antiviral drugs may be used to treat infections such as the herpes simplex virus. Acyclovir and femnocyclovir are popularly used drugs, derivatives of acyclovir, that are popularly used as well. Ganciclovir is another agent that's used. Ribavirin and Librivirin are over-the-counter antiviral preparations that you see in agents such as Abreva, which are used to treat fever blisters, which, as you know, are the herpes simplex type 1 virus, cold sores. So derivatives, all are derivatives of acyclovir, which was the standard for many, many years. Again, famciclovir and ganciclovir are probably the most popular agents that you'll see. The process by which these antiviral drugs work are basically to substitute in for nucleosides and nucleoside analogs. Again, they substitute for a nucleic acid, and in their substitution, when it comes to the process of transcription, the synthesis does not occur because the analog is there instead of the actual nucleic acid, and thus synthesis is arrested. Antiretroviral therapy. RNA viruses depend on what's referred to as the reverse transcriptase. We talked about this with HIV disease. HIV is an RNA virus, and antiviral groups is largely directed at that. In fact, the term antiviral implies that it's used against the HIV virus. Most of the antivirals are nucleoside analogs such as zidovudine or AZT, which is an analog to thymidine. Another one is cytofovir, which is used to treat cytomegalovirus that affects the eye. These infections are considered also for possible treatments for smallpox, but there's no definitive data in that as of yet. The only example of a nucleotide analog such as tenofovir is an antiviral that is a non-nucleoside agent, and this one is referred to as vipramine. Inhibitors of enzymes neuroramidase, zanamivir or Relenza, oseltamivir, phosphate or Tamiflu, used for the treatment of influenza. It's thought that oseltamivir or Tamiflu will be important therapy in treating the bird flu. Another approach to control HIV is the inhibition of the enzyme that controls the last stage of viral reproduction, which requires protease enzymes. Protease inhibitors such as enenavir or sanquinavir are examples of this therapy. Cells that infect a virus often produce interferon, which inhibits further spread of the infection. Interferons are basically cytokines. Alpha interferon, as we discussed earlier, is used in viral hepatitis infections. Newer drugs, imiquimod, stimulate the production of interferon. Now, again, this is an indirect effect on inhibiting viral growth. Again, the drug imiquimod basically stimulates the production of interferon. It's the interferon that will inhibit further spread of the infection. Categories of drugs used to treat HIV disease are protease inhibitors. We talked about those earlier. Indemnivir for HIV infections. Others to treat influenza, inhibit attachment of the virus to the host cells, such as zanamivir or Tamiflu, which is used to treat influenza. Amantadine, also used to treat influenza, inhibit uncoating of viral agents. And then interferon, which again is a cytokine, prevents the spread of viruses to new cells. So thus, it's felt like if you can keep the virus from spreading to other host cells, the virus eventually will die in the cell that it's infected, and thus you will arrest the spread of the virus. There are also drugs used to treat protozoans and helminths. Antiprotozoal drugs such as the quinines. Quinine is still limited use against malaria, but generally it's been replaced by newer synthetic drugs. Chloroquines and mefloquines are used now, and I won't say exclusively, because still some developing countries quinine is the drug of choice. Quinacridine is used against Giardia infections. It functions similarly to what we see in antiprotozoal drugs. There's also newer drugs, diodohydroxyquine, basically that's used as a drug to treat amoeba infections, and it's felt to be amoebicidal. Metronidazole, which we commonly see in clinical settings here in the United States, again is an antiprotozoal drug. It's used in treatment of many of the protozoal diseases. It's effective against certain anaerobic bacteria as well. It causes a disruption in the DNA under aerobic conditions. Again, antiprotozoal drugs include chloroquine, again diodohydroxyquine, which we see primarily used in the treatment of amoeba infections, and then metronidazole, which damages DNA, can be used in anti-amoeba and also trichomonas. Should be familiar with metronidazole. The trade name for metronidazole is Flagyl, F-L-A-G-Y-L. Should be familiar with this one as treatment for trichomonas, protozoal diseases. Let me go back. I think there may be another test question there. Yeah, you should also be familiar with quinine or chloroquine as early treatment for malaria. Specifically, anti-helminth drugs, most popularly used is mebendazole. And of the agents here, this is the one that you need to be familiar with. Mebendazole, treatment used to treat Ascaris infections. Now, there are other infections as well, from tapeworms, that use different agents, but the most popularly used drug for anti-helminths is mebendazole by and large. Again, the different agents, niclosamide, praziquantel, and pyantal, are drugs that used to treat helminth infections in one way or the other. We'll go through these in a lot more detail when we talk about microorganisms that attack the gastrointestinal system. The most popular drug used is mebendazole. Again, this is the one I want you to be familiar with. It inhibits nutrient absorptions in intestinal roundworms. Now, there are certain tests that we use as markers to be able to judge the effectiveness of certain drugs used in the treatment of a variety of microorganisms. So it's important to know how effective the drugs are that are being used. And there are certain studies that can be done to demonstrate that. One method is called the diffusion method. This is the disc diffusion method, or the Kirby-Bauer test. It uses a disc on an agar medium that's seated uniformly with a microorganism. When the antibiotic therapy is applied, it basically can demonstrate the effectiveness of the antibiotic therapy. These tests sometimes use a filter paper disc that's impregnated with a known concentration of a therapeutic agent. Now remember, this is typically an antibiotic. The chemotherapeutic agent, if it's effective when it's dropped on a plated agar dish that's covered with antibiotics, typically you see a zone of inhibition, or a clear zone where there would be no antimicrobial growth. The antibiotic that's been impregnated into this small little paper disc, this corn-sized disc, would be observed around the growth area. The diameter of the zone is used to calculate the susceptibility of organisms to an agent. There are more advanced studies that are done as well that help to estimate the minimum inhibitory concentration of organisms. Now, again, if you look at this plate, this agar dish has been plated with a microorganism. A lawn of bacteria has grown over this plate, and now what's happened? Little filters discs have been dropped. These filter discs are impregnated with antibiotic, and so if it's effective against this particular microbe, you'll see this zone of clearing where the antibiotic has killed the bacteria. Now, this is important because now we know this bacteria is sensitive, and this one is actually sensitive to each one of these drugs. However, the zone of clearing appears to be much more significant around this particular disc here, and if we can identify what this disc is, and again you see they're coded, so you'll know what antibiotic is impregnated in this disc. This is the drug of choice because it has a significantly large zone of clearing. This is another example. Now the E-test is a lot more specific. Why? Because it gives you a graded measurement and you know the MIC of these particular agents. MIC is important. Again, the minimum inhibitory concentration of a drug is known as the lowest concentration of an antibiotic that's used to prevent growth. If only a small concentration of an antibiotic is required, hopefully it means that that small concentration has less adverse side effects to the host cell. So we want the drug that has the minimum inhibitory concentration. The lower the MSC, the most effective antibiotic is determined by this agent. There's also what's referred to as a minimal bactericidal concentration. This is the lethal concentration that actually kills the bacteria, the minimum bactericidal concentration. Because remember, some agents are bacteriostatic, and at different concentrations an organism, a chemotherapy agent may be bacteriostatic, but not bactericidal. So the minimum bactericidal concentration is an important number to know as well. Many of these tests highly automated, and they use different techniques now, light scattering techniques, to determine whether bacteria is growing or not. So it's very automated now to be able to determine these concentrations. Drug resistance we've alluded to a little bit earlier. Resistance to a drug may be based upon the production of certain enzymes by organisms. Penicillinases produced by certain organisms inhibit the effect of penicillin. And if you recall, we talked about the use of beta-lactamases. Beta-lactamases can be made synthetically, added to certain drugs. With a beta-lactamase, the beta-lactamase will actually inhibit the penicillinase, and so the chemotherapeutic agent can still be used to treat these organisms. But resistance to drugs is basically based upon the production of the enzyme penicillinase. Another serious threat to chemotherapeutic drugs is resistance from various kinds of carrier plasmids. These are a little more difficult to control. Resistance factors, or R factors, plasmids, can be introduced into certain bacteria which will confer resistance. Plasmids may transfer genes for resistance to several antibiotics of that type. So this is done at a genetic level. It's closely related to bacterial species. Bacteria that drugs are unable to control control of certain superbugs which have typically these resistant factors, and these superbugs may be polydrug resistant as well. Another test that helps to demonstrate the concentration of antibiotic therapy that's needed to be used to treat certain drugs helps to determine the MIC and the MBC. Now, for antibiotic resistance, there are a variety of mutations that can lead to resistance. The mechanism of antibiotic resistance typically has four sites: enzymatic destruction of the drug, prevention of penetration of the drug, alteration in the drug's target, or rapid ejection of the drug. Resistant genes are often plasmids. Again, this is at a genetic level. Misuse of antibiotics help to also form resistant mutants. Using an outdated antibiotic which is weakened and not completely effective allows the microorganism to develop resistance against it. Using antibiotics for a common cold when really you're only treating a viral infection. Remember, antibiotics are only used to treat bacterial infections. You would need an antiviral agent to treat the common cold, which is a common etiology for colds. By giving an antibiotic when it's not required actually allows microorganisms to develop resistance. The use of antibiotics in animal feed affects the host as well. This is because we typically eat the animal flesh, thus we do get concentrations of antibiotics in our own system. The bacteria that are a part of our environment will eventually develop a resistance as well. Failure to complete prescribed antibiotic therapy. What happens in this case, we start taking antibiotic therapy and we take it until we feel better. We may not have eradicated all of the bacteria. We have not reached the concentration that's bactericidal, but we certainly killed a significant amount of the bacteria. However, we did not completely eradicate the infection. The remaining bacteria in our system now has been exposed to the antibiotic and will develop resistance. Thus, when it comes back newer and stronger. So it's always important to complete antibiotic regimens. And then, of course, if you use someone else's leftover antibiotic prescription, the person may not have been on a broad-spectrum antibiotic. They may have had a specific type of infection that was treating a gram-negative organism. You have a gram-positive infection. The only thing you've done by taking their antibiotic therapy is to develop a resistant organism. Many drugs you'll see, especially antibiotic therapy, are used in combination. You see two drugs given simultaneously can be more effective than either drug given alone. This is called synergism. Other combinations can show antagonism, which is the effect of two drugs is less than that of either one used alone. Synergism occurs when the effect of two drugs are used together, which have a greater effect than either alone. Examples are the use of an aminoglycoside with a narrow spectrum antibiotic, such as penicillin given with gentamicin. Antagonism occurs when the effect of two drugs together is less than the effect of alone. So you need to make sure that there's not a drug-drug interaction when the person is taking more than one drug. Again, the future of chemotherapeutic agents look at antimicrobials that have broad-spectrum antibiotics. They can be derived from plants or animals. There are other antisense agents, which basically are complementary DNA or peptide nucleoside acids that bind to the pathogen's virulence genes and helps prevent transcription. The most pressing concern currently is the spread of resistance to antibiotics or the spread of resistant microorganisms. Antimicrobial peptides, such as in plants, insects, many animals, defend themselves against microbes using broad-spectrum antimicrobial peptides, sometimes called cationic peptides. There are some examples of these, the mecanines, the squalamines. I'm not going into any detail about these, but these are just different antimicrobial peptides that are out there on the market. A lot of research are done now. There are certain types of drugs that are used as food preservatives that resemble mode of action of some of these, but again, these are still being studied as agents to use. Antisense agents of specific importance, another approach to microbial control, are these short-chain synthetic agents. These are antisense agents. Principally, the principle is to identify sites in the DNA and RNA of pathogens that are responsible for pathogenic effect. And if you can attack these sites with these antisense agents, you can inhibit microbial growth. We also see antiviral therapies based on the same principle. Some have already been used in the treatment of certain antiviral agents, such as cytomegalovirus that we see in retinitis. Retinitis is